All right, and this is cellular reproduction, part 2. And today we're going to be looking at specifically the events involved in mitosis. No, mitosis is not a disease. Mitosis is division of the nucleus. Information here should be able to be directly put into your projects for explaining mitosis in your own words. Anyway, here it goes. All right, you already know that mitosis is going to be the main way we're going to just get new cells, replacement cells, cells that are identical to the original parent, should be able to do everything that the parent cell does. They're also identical to each other. So if we want to just make new cells, this is what we're looking at. Now, for us, if we had been starting with a zygote, and this would be a diploid cell dividing so that eventually we get diploid daughter cells. And this is the way we all start out. You need to go ahead and describe everything that's happening in the phases. Now remember, the vast majority of a cell's life cycle is going to be spent in interphase. And this is not a resting phase. This is where the cell is doing all of its work. Yes, it's going to prepare to divide, but cells do more than just reproduce. Cells have a particular function, and it's during interphase where they pursue all those particular functions. You will go ahead and be required to write the events of interphase and include those in your project. So even though interphase is not technically one of the phases of mitosis, you need to go and include it in your descriptions. Now, you will have to go ahead and not only sketch interphase, but you'll have to be able to learn how to recognize it. The hallmarks that you use for recognizing any of the phases are always going to be written in red in this presentation. So that's what you're going to be looking for, and that's what you're going to write down in terms of, hey, how can you recognize this phase? The events, the specific events that happen, well, those are always going to be written in green. Those also have to be included along with your sketch. Now, the G1 stage, that first growth stage, you can consider that period of time where the cell has divided, and now you have two smaller daughter cells. They're smaller than the original parent, so they have to grow up to about the same size as the original parent. And this is happening in G1. Of course, cells can't get big forever. There's a limit to how much they can grow. Remember, they have a kind of a packaging problem. They can't get material into or out of the cells fast enough, so eventually they have to divide. So once they reach their maximum growth, chromatin in the nucleus will start to duplicate. Now, although you're not going to be able to see this with the light microscope, this has to happen before the nucleus can duplicate. All of the genetic information, of course, is packaged in the chromatin. Once that is accomplished, then, yeah, the cell has to assemble structures that you're going to need for the rest of the division of the nucleus, mitosis, and the division of the cell, cytokinesis. Okay, here's an animal cell. This is from a developing fish. This one's in interphase. And note that you can clearly see a cell membrane, but you can also see a nuclear membrane. All right, so the nucleus is intact, and although you see a nucleolus, little dark areas in here, you can't really see any chromosomes because they're arranged still as chromatin. So the hallmarks you're looking for, hey, there's a clear nuclear membrane, well-defined nucleus, but you can't see chromosomes. And here we have a plant cell. This is from the root root tip of an onion, and also an interphase, so you're looking for the same distinguishing characteristics. You can see a definite, well-defined nucleus with a nuclear membrane. You might see dark splotch inside the nucleus, the nucleolus, but again, no chromosomes. The nuclear material or the genetic material is existing as chromatin, too thin to actually go ahead and see. After G2 is completed in interphase, cells ready for mitosis. And the first phase of mitosis involves prophase. Right? The chromatin has already been duplicated. The cell's got everything it needs to go ahead and divide. So division of the nucleus is actually going to occur. The chromatin is going to shorten and thicken, eventually going to condense into worm-like structures that you could actually see, the chromosomes. When you start to see chromosomes, you know you're in prophase. In animal cells, centrioles, little microtubules, or made from microtubules, will appear, and at the end of this phase, the centrioles are going to migrate to opposite poles of the cell, with the chromosomes all piled up in a great big ball in the middle. In this view, you've got two animal cells in prophase. The one on the left, you can still see the chromosomes. You cannot see a nuclear membrane, and you can't see individual chromatids joined by a centromere or anything like that, because the chromosomes are just tangled together in a ball. The cell to its right, well, same thing, but you got this little dark structure out here. Well, that's most likely the centrioles starting to form. And eventually what's going to happen is they're going to go ahead and separate and move to opposite ends of the pole. One on this side and one on that side. But that's not till the end of this phase. Plant prophase, very, very similar. Again, the chromosomes bundled together, tangled in a ball in the center of the cell. You cannot see a nuclear membrane. It's been dissolved and disintegrated. What you're not going to see here is centrioles. Plant cells don't have centrioles, only in animals. Meta means middle. So here, we're going to have little microtubules called spindle fibers, and they're going to kind of pull the chromosomes into place so that they'll line up in the center, but instead of being in a tangled mass, a tangled ball, they're going to be kind of sort of lined up almost single file. And right along the central portion of the cell, you can think of this as the cell's equatorial plane, almost like the equator of the globe. A good view of metaphase in animals. Note that the chromosomes, if you were to go ahead and draw a line through them, you could probably hit almost all the chromosomes with a straight line, assuming I could draw a straight line. Centriole, here and here, and radiated out from the centriole would be those spindle fibers kind of sort of attaching to the chromosomes. Hey, same thing in a plant cell. You got a plant cell in metaphase on this side. These puppies, well, you can't see any chromosomes, but you can see a cell membrane, so I'll let you figure out what phase those are in. Chromosomes are clearly visible in a straight line, kind of sort of lined up in the middle. And here's a good view of some spindle fibers. Right here, right here. Now in animal cells, the spindle fibers are running from the centromere of the chromosome to the centriole. Here, the spindle fibers are kind of sort of wrapped around each other, right there and right there, but there is no centriole. Okay? Again, metaphase, you can recognize this one because the chromosomes are all lined up in a row, right there in the center of the cell. All right, anaphase. Now, here's where some real action occurs, and what allows for this is that the centromeres, the center of each chromosome, the centromeres will duplicate themselves. Now each chromatid is going to have its own centromere. So for a brief period of time right now, we have twice as many of chromosomes. Remember, you define a chromosome by number of centromeres. The spindle fibers will actually shorten. And as they shorten, they're going to pull the now chromosomes to either the left or the right half of the cell, or the north and south portion of the cell, whichever way you want to look at it. But you're going to have two distinct sets of chromosomes going from point A to point B. So once again, in anaphase, we have two sets of chromosomes, and that's what you're looking for. That's our big hint that you're in anaphase. Two sets of chromosomes, and this set is going in this direction, this one's going in that direction. The other thing that you might want to look for is trace the edges of the cell. There's no sign in the edges of the cell membrane, that the cell is undergoing any cytokinesis. You still have one cell, but two distinct groups of chromosomes. No difference in the plants. Two sets of chromosomes, one moving in this direction, the other moving in that direction, and no signs of cell division. So a good anaphase in plants. Telophase is our final stage, and you're still looking at two sets of chromosomes, so sometimes this can be confused with anaphase. But what you're really looking for is that telophase kind of sort of coincides with cytokinesis, the actual division of the cell. So if you see two groups of chromosomes, but the cell actually starting to divide, then you're in telophase. Now, the chromosomes are arriving at opposite ends of the cell. When they do that, the actual nuclear membrane is going to reform, and the chromosomes are going to kind of stretch out, turn back into chromatin, and the cell's going to get ready for another interphase. All right, so here's a really late telophase here. And set of chromosomes here, set of chromosomes here. And the reason I'd call this telophase, and actually really, really late telophase, is that if we trace the nuclear or the cell membrane all the way over here, or all the way over here, or all the way over here, versus all the way over here, there's just a narrow place where these cells are still connected. So these ones have almost completely and totally divided. Right? So telophase for sure. Now in animal cells, yeah, they can kind of sort of pinch off to form new cells. In plants, you got that cell wall to worry about. So what you'll have is a cell plate forming right there between the massive chromosomes. And that's going to be the new cell wall that's going to develop between the two new cells. Here's another late telophase. It's hard to even see chromosomes in that because the nuclear membrane is already starting to reform. But either way, in animal cells, hey, look for that pinching off. In plant cells, look for the formation of a new cell plate. Those events will go ahead and signify the beginnings of cytokinesis. And when cytokinesis starts, hey, you know you're in telophase. This is kind of a neat view. This is using a technique called fluoroscopy, and it shows the various phases that we just went through, plus a little bit more. Note the differences between what you see in interphase in terms of the nucleus versus what you're seeing in prophase. We do see the beginnings of some chromosomes, and also here, right there, you got a couple of centrioles that look like they're starting. The reason this is called pro-metaphase over on the right side, this is not a something where you just flip a switch and you go from one phase to another. This is actually a continuous process. So we're kind of sort of proceeding from prophase on into metaphase here. And you're kind of like in between the two. Not quite in metaphase yet, but certainly late prophase as those spindle fibers start to form and those chromosomes get pushed into place. If you had difficulty in the last slide looking at or envisioning what was going on, here's an artist's conception of what's going on. So you can see these things in a little bit more detail and kind of sort of cleaned up. But exact same view, just kind of an artist's conception. Hey, and here's the rest of it. A definite metaphase over here. You can see in the chromosomes outlined in blue, the spindles are in green, and you can still see the centrioles there serving as anchor points. And they'll separate again in anaphase, and a good view of cytokinesis going on, that pinching off process in telophase. And just as before, an artist's conception of what's going on. Right? By the way, you can think of these centrioles almost like anchor points. So when the spindle fibers shorten, right, they're not going to move from their anchor point. So as they shorten, the only thing that can happen is that the chromosomes are going to be pulled in one direction or another. All right, this is kind of a cool slide to practice on. So you can grab this with one of your friends and go ahead and pick a cell, any cell, and you should be able to go ahead and have them tell you what phase it's in, and more importantly, why. For instance, the one that's circled there in green should be in metaphase because they're all lined up in a center. But hey, pick others, any others, and hey, how about this one in red? Or maybe this one in blue. Or, dude, how about this yeller one right there? Anyway, give it a try. These are things to practice on. These are things that you should be able to do by inspection. Hey, good luck.